Well, welcome back to the Kavli Prize 2018, and now we're celebrating the second. We go from the largest, astrophysics, to the very smallest, nanoscience. Now, over the last 5 years, the Nanoscience Prize has generally gone to some pretty hardcore physics, as the name implies, nanoscience. But when I discovered that the Nano Prize judges this year had awarded the Kavli Prize for nanoscience to 3 geneticists, I am a geneticist, I was extremely excited because finally we were talking about a nanoscience that I could understand. Now, we will talk about genetic engineering, which is something that has been part of our lives now for 30 or 40 years, but in the last few years, no more than 10 at best, a new technology that was invented by our winners, our laureates today, has come on the scene and has I'm slightly allergic to hyperbole in science and in science reporting, but it isn't an understatement to say that CRISPR-Cas9 technology has entirely revolutionized almost every aspect of the biological sciences and is set to continue to do so for the future. The repercussions for science and for society cannot be understated, and it is for that reason that the Nanoscience Prize, the Kavli Prize for 2018, has gone to 3 geneticists, Virginijus, Jennifer Doudna, and Emmanuelle Charpentier. Now let's have a look at their work.
Many big discoveries in molecular biology happened by trying to find answers to very basic biological questions. And the very basic biological question that we are trying to address in my lab actually is how bacteria defend themselves against viruses and how different antiviral defense systems in bacteria function. We look at molecular mechanisms. We are trying to understand what are molecules and proteins in bacteria that actually provide this interference against invading viruses.
Microbiology has really been at the origin of the CRISPR-Cas9 technology, because this is through the understanding of certain mechanisms existing in bacteria and also existing in viruses that we could develop all the technologies that have been used over the last 50 years in molecular biology and also in genetics.
CRISPR is an acronym that stands for Clusters of Regularly Interspaced Short Palindromic Repeats. It's a big mouthful, but basically what it means is a place in the bacterial DNA where cells are able to store genetic sequences that come from viruses. So they keep a genetic record of past infection. It's a fantastic way that bugs can acquire immunity to viruses. And it also turns out to be a powerful technology for controlling genomes, because the enzymes, the proteins that are part of these CRISPR systems, are effectively programmable DNA cutting proteins.
In a simple way, CRISPR-Cas9 is to be seen as a kind of scissors that are programmed to recognize a certain sequence on the genome of any type of cells and organisms. And these scissors will cleave the DNA, and this will trigger genome modification at the site expected to be modified on the genome.
I think what makes the CRISPR-Cas9 technology unique and truly revolutionary is the way it works. It's a technology that works by recognizing a short sequence of DNA using a chemical copy of DNA called RNA. And cells will naturally make RNA molecules that program the Cas9 protein to find and destroy viral DNA. Scientists, now that we understand how it works, we can actually make our own pieces of RNA that direct Cas9 to any desired DNA sequence.
Now there is for sure a tremendous potential in just research and development to use genome editing to understand the functions of genes in human cells, to be able to develop novel anti-infectives by finding new targets for therapeutics, by developing models of diseases that are more clinically relevant.
Cas9 could be used to correct mistakes in the DNA that cause genetic disease, and this could help us to actually cure genetic disease that were incurable for a long time. Cas9 technology also can allow us to introduce different changes in the DNA. This is important also in agrobio field, because new plant varieties could be engineered that actually could be resistant for pests or droughts or other environmental factors.
And it's easy to do, so it makes it trivial to make new tools for any part of the genome where a change is desired.
I really do hope that with regard to the CRISPR-Cas9 technology, that it will reveal to be successful to treat at least certain human genetic disorders, and that it will have really brought really a plus in the treatment, whether it's by with a direct application or indirect application in the overall field of biomedicine, really for the treatment of various types of diseases.
Now, Jennifer Doudna will be joining us later today, but please welcome to the stage the Kavli 2018 laureates for Nanoscience, two geneticists, Virginijus and Emmanuelle Charpentier. Okay, well, first of all, congratulations. It's very exciting for me as a geneticist to see two geneticists on stage with me for Nanoscience. Can we start off by just talking about the technique for a second, because as I mentioned, we've been doing genetic engineering now for, well, since the 1970s, and it is part of many people's everyday lives. But this is different. CRISPR is a new technique, and it is transformational in terms of genetic engineering. Why? What is different about it?
What is different about CRISPR-Cas9 is the fact that it's a very versatile technology. It really allows to edit the genomes in ways that were not possible prior to CRISPR-Cas9. And it's a very powerful technology. So in principle, it is true that us biologists were able to modify genes and their expression for about 50 years now. However, the tools that were available prior to the possibility to do genome editing that started actually prior to CRISPR-Cas9 with some enzymes named zinc finger nucleases and talon nucleases, but before the possibility to do genome editing, there was only the possibility mainly to do genome recombination. And also with regard to, for example, working with certain organisms, limited to work, for example, with regard to higher eukaryotes, if one wanted to understand functions of genes in human cells, had to go to the mouse model, because in human cells, either it was not possible to really act directly on the DNA, or at some point it was possible to act via RNA interference technologies on the expression of the DNA, but this was not that clean. So in principle, CRISPR-Cas9 brings also the possibility to do genetics in organisms where it was not possible before to do genetics.
So it's the speed at which it can be done?
Yeah.
So 10 years ago, it took me 2 years to do some genetic engineering on mice, and at the end of it, it failed. That can now be done in about 2 weeks by an undergraduate.
Exactly. Which
Put
is
you back in your box.
Indeed, the point which also reminds me my time in the US when I did a postdoc and focusing on transgenic mouse models, and I spent more time engineering my transgenic mice than analyzing the phenotypes of my transgenic mice. So I think now it's a little bit easier. Having said this, it's important to say that gene editing was existing prior to CRISPR-Cas9 with two types of enzymes, as I said, zinc finger nucleases and talon nucleases, that actually act, at the end of the day, they do exactly what CRISPR-Cas9 is doing, but the mechanism used is more cumbersome. The way to design the technology was more difficult. It was taking more time. but gene editing, so the ability to really introduce mutations at a certain location on the chromosome, to correct mutations, to introduce new pieces of DNA at a certain site on the chromosome, or to exchange a piece of DNA by another piece of DNA, to exchange a gene by a gene was possible prior to CRISPR-Cas9. But CRISPR-Cas9 brings even more than this.
It does.
It's such a versatile technology thanks to the very simple mechanism existing in bacterial CRISPR-Cas9, yet sophisticated enough to bring this potential of versatility. So to act on the DNA, to act on the expression of the DNA, to also act on the RNA, and to have also, it's a tool that can be delivered not in a complicated manner, so allowing really to...
The diversity is really important as well for its widespread application, because we've been talking about mouse models for human diseases, and of course, of the model animals that biologists use, there are a few that are very well characterized genetically, and we can do traditional genetic engineering, and with CRISPR, you can effectively apply it to almost any organism.
Yes. And even Drosophila, Drosophila has been really a model that has been studied extensively, in which genetics was possible. CRISPR-Cas9 brings also a level of simplicity. So scientists working on Drosophila are very happy to have CRISPR-Cas9 because they can do more precise genetics.
It's the precision, isn't it? It's that you can change as little as one base pair.
The precision and the efficacy of the technology.
Yeah. Are you, in the last few years, have you been surprised by the uptake of CRISPR by pretty much every molecular biology lab on Earth?
Yes and no, actually. The reason also why I focused on CRISPR-Cas in my lab at a certain time when we were working on different topics, it's because I knew that there was some, let's put it that way, some lack of efficient genetic tools specifically for eukaryotic systems. Because myself, I mostly worked on bacteria, and I'm a bacterial geneticist forever, but I worked also on eukaryotic system at some point, enough to realize that actually the tools were missing. And I work on bacteria causing diseases in humans, and I was interested principally to learn more about eukaryotic biology, to really study more precisely host-pathogen interaction, realizing that the bacterial pathogen I'm working on infects really human cells. And then at the time, so 20 years ago when I was a postdoc in the US, there was no possibility to really edit the genomes of human cells, so not really the possibility to study properly the bacterium within its natural environment. So I was aware of the fact that tools were missing, and so that CRISPR-Cas9, among the different CRISPR-Cas systems existing, may provide at least a simple mechanism that will be useful to the biologists. What I underestimated is the fact that really the biologists were in need of such a tool, and that beyond applications which I was thinking of, which human cells, plant cells, and certain model organisms, that it allows also really the genetics in more organisms which were not possible to manipulate genetically before, which were a little bit difficult to manipulate genetically before. And this is what I like with CRISPR-Cas, is that it originates from evolution of bacteria. It's a bacterial mechanism originally that has largely evolved. And it allows to provide a technology that allows to really study the diversity of the world and the evolution of the world, and to really study the right types of cells which are relevant for the biological questions which scientists want to ask.
So Virginijus, this came from a really, I mean, it's been under our noses for a very long time, and it wasn't until the mid-1980s that somebody spotted this very odd pattern of these palindromic repeats in bacteria. And you at the time were working on nucleases, on restriction enzymes, which is another method of trying to be a sniffer dog, if you like, for particular sections of DNA. And you read that paper that came out in 2007, and you instantly gave up your restriction enzymes and went into Cas9. What made you so excited when you read that paper?
Actually, I was working on molecular scissors for most of my life. So when I was doing my PhD work, so I worked on molecular scissors that cut proteins and help us to digest food. And then I switched to other type of molecular scissors that are called restriction enzymes that actually, as you said, they recognize short nucleotide sequences that cut DNA very, very precisely. And I was trying to understand actually the unique specificity of these restriction enzymes for many years. But at the end, spending nearly 20 years in the field of restriction enzymes, I was looking for something new and was thinking about changing fields. And then this paper appeared in Science in 2007, and which actually carried an CRISPR acronym that is nearly as unpronounceable as my family name. So I thought, oh, this is probably an interesting system, and no one knew how it works, so that I decided to jump on it. Yeah.
And then you were completely hooked.
Yes, exactly. So that actually I'm hooked until now.
And the Kavli prizes are a great celebration of basic research, although there are obviously huge implications for the applied technology that is CRISPR-Cas9. But when you were looking in bacteria, when you were looking at this system, just give us a flavor, where they mention it in the film, about what it's doing, why it evolved in the first place.
Actually, you know that actually we have immune system that protects our bodies against pathogenic microorganisms that are present in our environment, and probably we are less aware that actually bacteria also have their enemies, and these enemies are called phages. And they actually are the major parasites of bacteria, and bacteria survive because they have multiple layers of defense that form a kind of very primitive immune system of bacteria. And as you know, that actually the immune system is comprised of two branches, like an innate immunity and adaptive immunity. And adaptive immunity actually is able to memorize invaders. And the CRISPR system actually functions as an adaptive immunity system in bacteria, because actually it can recognize the invader just putting a small piece of invader's DNA into their own genomes, and then use this information to recognize the invader in the next round of infection, and use this information then to recognize the invader and degrade it.
So it's a bit like a passport stamp. You get a stamp in your passport, you stuff it in your DNA if you're a bacteria, and then the next time that phage comes along that's similar, you've got the weapon. First of all, you know that's the phage, and then you need to roll out your defenses.
Yes, exactly. It's like fingerprinting of the phage and putting it in the kind of pocket, and then checking next time whether it's the same guy or not.
And in terms of developing this into a tool that now we're all using, how do we get from the short version, please? How do we get from the observation that this is a system that bacteria have into this is a kit you can buy from a company, and first-year undergraduates here in Oslo are using it?
Can you get it on Amazon?
Probably.
I did not check. No, actually, this is the beauty of the CRISPR-Cas9 mechanism, is that it works in such a way that the harnessing of the mechanism into the technology was quite simple. And actually, we can use the natural mechanism that is actually an enzyme guided by a duplex of RNAs to really use this mechanism directly as a technology. So the only thing that the biologist has to do is to design the RNA molecule according to the sequence to modify, and for sure some tricks which geneticists know how to design. But yes, this is really the beauty of CRISPR-Cas9. Is that
I mean, it is almost as simple as that, isn't it? You know if you know the sequence of DNA that you wish to edit, you design an RNA molecule which represents that.
Yes.
And you send that off, and what you get back is a kit that'll cut it out. Ish.
Yes, principle. Yeah. I mean, it's a bit more complex than that.
A little bit.
But it's...
Yeah. And in my life, BC, it means before CRISPR. So
I was trying to engineer restriction enzymes to cut any DNA sequence, but it was tough because actually to engineer restriction enzyme to cut different DNA sequences, you need to engineer protein, and this is tough because you need to do a lot of protein engineering.
Yeah, which is
and the beauty of CRISPR is that actually you don't do protein engineering, you just change RNA molecule, and that gives you really very nice programmability of the system.
Yeah, which is again another reason why it's been taken up around the world with such enthusiasm. Now,
but the idea that these CRISPR-Cas systems involve RNA-guided enzymes that can act on the RNA and mostly DNA. So this was one thing to think that one could exploit such system for genetic tools, but colleagues of us actually were working on CRISPR-Cas systems that were quite complex, where it was a bit difficult to imagine that such system could be harnessed into powerful and easy technology. And CRISPR-Cas9 made a difference because it's an enzyme guided by a duplex of RNA. So it's a three-component system, and so one had to understand the mechanism, which we did, and therefore
we've been talking about how this has come out of basic science. And we'll talk a bit about the applications and some of the ethical implications, but let's keep on the track of basic science for a moment. What discoveries do you think might be possible with CRISPR-Cas9? So, we talk about it mainly as an application for, the treatment of disease and that kind of thing, but it's also an extraordinary tool for discovery. So what kind of discoveries do you think might be possible?
Understand better the mechanisms of life, most likely question certain dogmas, because now there is a possibility to actually act on the DNA in a more precise manner and in more types of cells. So also realizing that, as often it is when one goes deeper, is that certain mechanisms which one was thinking that this will be the way a mechanism works in every type of cells, that cells have also evolved certain diversities of mechanisms.
So not just BC, but we're rewriting the gospel according to CRISPR now with the new technologies.
Yeah,
this analogy may go too far.
Yes.
Actually, and CRISPR are applied in very, very different fields, starting from basic biology to answer very basic biology questions and to end up with biotechnological applications. And if you Google, you can find also like a link that says most CRISPR applications. So that's also gives you an idea that actually there are multiple layers of CRISPR applications in very different fields, and I think that CRISPR application in development biology is very important. That helps us to understand the very beginning of the life.
Yeah, well, that's my field, and that's why I'm super proud to be on stage with you, but also kind of a bit annoyed that you didn't do this 10 years ago. It made my life entirely different. Let's talk about humans and gene editing in just a minute, but before we get to that, because there are obviously many people have interesting ethical questions about gene editing for humans, but let's just talk about some of the subjects that don't get covered in the press so often, which aren't to do with humans. For example, my understanding is that CRISPR has enormous potential for incredible impact in the field of conservation biology. Nod if you agree.
What do you mean by conservation biology?
Well, understanding genetic diversity in species that are endangered. God, you look blank for a second.
Yes, this is like the kakapo. So the kakapo, the most stupid bird in the world, which is now down to about, how many is it?
165.
165.
Do you know the kakapo? Are you aware of the kakapo as a
New Zealand bird that eats
yeah.
windscreen wipers?
Yeah.
Yes, it was almost extinct, down to 55 birds. New Zealand scientists told me this when I was out there last year. There are more kakapo alive. No, they're not. There are more kakapo in museums in Europe than there are alive in the wild. But the problem is when a species becomes lacking in that size, it lacks genetic diversity, and it is effectively extinct anyway. So they had the idea of sequencing the genomes of the kakapo in museums around Europe from a time when their population was healthy, and then CRISPRing in to the live birds a new level of genetic diversity in order to rescue them. Now, this is pretty pie-in-the-sky stuff, but that's an astonishing ability to be able to do that, entirely enabled by your tool.
I think it was,
Could you do that?
I think it was maybe possible even prior to CRISPR-Cas9, maybe not in such a precise way. What I want to say by this is that those questions of manipulating the diversity and the evolution was possible prior to CRISPR-Cas9.
But not with the accuracy we can do.
Not with the accuracy, indeed.
And that's essential. What about food production and crop development? That's another thing that CRISPR is going to radically change, or in fact is already radically changing.
Yeah, so exactly. CRISPR gives a really huge opportunities for plant technology, because actually it allows to speed up selection procedures really immensely, because actually then you can introduce targeted mutations at genes where you like to introduce mutations, and to do it much more precisely than people are doing right now using technologies that are available now.
Do you think people will be concerned about the use of CRISPR in food plants and food animals in the same way that they were about GMOs in Europe? Because we had a time when people became very agitated about the use of genetic modification in plants, and there was a complete moratorium effectively on research in genetically modified crops in Europe for the best part of a decade because of that. Do you worry that that might be an outcome of CRISPR use in plants, food plants and animals?
Yeah, but in fact, genetic modification of plants is already going for ages, and people are just trying to select different plant varieties or introduce new different plant varieties using different tools. And in fact, now there is a major debate that is going on that actually, and there are two schools of thought. So one school of thought is thinking that actually genetic modification is dependent on techniques, what they are doing in plants, and another school of thought says that actually we should look at the product. And actually, if you look at the product, so then actually modifications that you obtain by other conventional techniques and modifications that you obtain using CRISPR technology, they are indistinguishable.
So it's a tool, and when we look at these ethical questions, you should look at them on a case-by-case basis rather than saying this is the technology that we should ban, if that's what people are talking about.
Now, if Jennifer was here, I know that she'd talk about the ethical implications of CRISPR, and she's put a huge amount of effort into talking about it and starting public debates about the use of CRISPR technologies in humans. And it's a very attractive thought that you might be able to somehow prevent disease. I mean, first at a somatic level, in other words, cells that are not inherited, but even so, even though CRISPR is very accurate in itself, there's still the problem of delivery, in the same way that actually there's been a problem with delivery in all types of genetic engineering. Are there any more issues about off-target effects with CRISPR than there are with other technologies?
No, I think that actually CRISPR technology is quite a precise technology, and of course these off-targets happens, but I think that we should think about the importance of off-targets, and we should clearly differentiate fields, because actually if we talk about plant science, actually it's not a big issue of targets, because these off-targets cannot be outcrossed, and it's of course a bigger problem when we talk about human engineering of using CRISPR for engineering of somatic cells in humans.
I want to talk about a little bit the culture of science, and when we're talking about the ethics in general, and I'm sure the audience will have questions which we'll come to in just a minute. But what has it been like for you guys over the last few years, being at the center of a genuinely revolutionary technology which is now used by every lab on Earth, and also the press have a complete feeding frenzy about every single CRISPR discovery, and people want to know about it. What is it like being a scientist, basic scientists, at the epicenter of that sort of storm? Emmanuelle.
In which context?
Well,
What's your life like? We're sitting on stage in front of all these great Norwegian people.
I guess you don't want to know what my life is. But so with regard to ethics, I have to say that, no, because it's quite complex. With regard to the fact that indeed it takes a lot of my time, CRISPR.
We'll let you go in about half an hour.
Yeah, no. Having said this, I'm glad to have witnessed a number of discussions happening in Europe, mainly through actually really the work of many of my colleagues at the National Academy of Sciences. So maybe it's less, let's put it that way, in the media than the discussion that have been going on in the US. I've also been involved in discussions with politicians at the parliament in Germany, and also discussing with députés and membres du Sénat of France. And there is really a wish for the politicians to understand CRISPR, the technology and the implications of the technology, because there is for sure a responsibility of everyone with regard to the applications, what we discussed with regard to food and also human medicine. So I'm quite happy to have witnessed all those discussions. Now, surely there are also differences with regard to cultures and views of how far one can go with regard to the applications of CRISPR-Cas9. So Europe is more conservative, with for sure in Europe, the UK and maybe Nordic countries being more liberal than France and Germany, for example. So you may have heard about the recent decision from the European Court of Justice to the decision that is that a plant engineered with CRISPR is considered as a GMO, which was a bit surprising. So there are more restrictions in Europe. There are also restrictions with regard to the development of the technology to modify the genomes of human germlines. Also some restrictions with regard to the usage of the technology to modify human embryonic cells, even if it's for research purposes. For example, there are certain countries which will ban this kind of research. In the
we're
US,
beginning
it's
to
more
see
liberal.
that those papers are coming out of China
Yes.
more prevalently than anywhere else at the moment. Is
Yes.
that something that
Yes.
we're
And
very
I think
aware
in the
of?
US also it's a little bit more, I think, liberal. There are some how do you say, statements from the National Academy of Sciences that are, let's say, if you read between the lines, could open the door in the future of certain applications.
Because what happens when a new technique comes out like this, and we're already seeing it, is you see an absolute explosion of use. And actually not all of it is great science, is it? I mean, I guess people are just trying the technology. Do you think that the field will then settle down, Virginijus, so it's then more considered?
Yeah, so, but if you look backwards, so then actually in 1980s, so when genetic engineering appeared, so when people learned how to cut DNA and how to clone DNA and then actually how to engineer microorganisms, there were also worries that actually it will be a problem that actually scientists will engineer superbugs that may kill humankind. But it didn't happen. And if you look retrospectively, so the technology was tamed and actually it produced benefits for humanity, just making new proteins or curing diseases. So I think that the CRISPR will follow the same path.
Okay, let's open it up to the audience. So if we could bring the lights up now.
Thank you very much. My name is Hans-Peter Graver. I'm a professor at the law faculty. I just wanted to ask you about your reactions to the ruling by the European Court of Justice. Do you believe that the court has misunderstood the CRISPR technology? And in case, in what way has it misunderstood it?
So just to give some context, there was a question from a lawyer.
Yes.
Which refers to the recent ruling by the European Court of Justice that plants that have been CRISPRed, are we allowed to use CRISPR as a verb?
Yes.
Yeah, okay. That have been modified, that have been CRISPRed, should be subject to the same rules as genetically modified organisms done in the traditional way. Now, the question was, do you feel that the European Court of Justice has misunderstood the technology?
I believe so, indeed, that it has misunderstood the technology. As far as I know, the definition of GMO as of today is an organism that would have been modified genetically, whereby at the end of the process there has been introduction of foreign DNA, which is not the case for a plant that would have been engineered with CRISPR. I mean, one can introduce foreign DNA, but one can also CRISPRize the plant, whereby at the end of the process there is just maybe one single nucleotide that has been modified, and in such a way where there will not be any difference between a natural plant, a naturally engineered plant, and a CRISPR engineered plant. So I believe that there has been maybe some misunderstanding indeed with regard to what the technology brings in terms of advantages of really the production of clean plants, clean crops.
And actually, I think that European Court of Justice stick to the regulation of technology instead of looking at the product. And then when you stick to regulation of technology, so you have an inner problem when technology changes. So then actually really difficult to apply the same set of regulations to a new technology. So that's
And the repercussions after that ruling, which was only in the last couple of weeks, or last month, I think, do people come to you and ask you about these questions? Is there a process by which that ruling might be challenged by scientists who are experts in the field?
We are experts in a scientific field, but actually we're not lawyers, and it's difficult to answer this question. Yeah.
Okay. There are some concerns indeed from among scientists with regard to this decision. Now I'm not so sure how this can, be reversed, or let's say in a short.
But you gave a very clear answer on that. You said you thought that they had misunderstood the technology, and I suppose the fact that we're having this conversation is part of the public discourse about challenging it. So we
not only the technology, but also the implication of the technology for the production of crops. So the technology itself, and also what the positive aspect that it could bring in food production, with regard to all the issues of climate changes and.
Okay.
Let's take another question.
Hi, my name is Sigrid Bratteli. I'm with the Norwegian Biotechnology Advisory Board. Now we're getting closer to application of CRISPR in clinical use for humans in somatic treatment of diseases. So can you say a little bit about where you see this going in the next 5 years or so? Where are we in about 5 years' time in terms of clinical use?
Okay, so the question is, in the next 5 years or so, where do we see the application of CRISPR technology for clinical use, potential clinical use for diseases, which I guess is, fundamentally your interest in the subject?
Mm-hmm. So first of all, we have the indirect application of CRISPR-Cas9, in the sense that CRISPR-Cas9 is helping a lot the field of human medicine by really boosting research and development. So all those screens that are used to find new targets for therapeutics, the technology that is used to develop models of diseases that are more relevant to the clinical situation and allow to really understand better the causes of diseases. So this is one thing, and this for sure it has already evolved. I mean, we see more and more publications coming up with regard to this aspect. Now, as a direct medicine in principle, or more direct use, you have the use of CRISPR-Cas9 in cancer immunotherapy, and this you have some clinical trials that have started, and hopefully one will see some success in the next 5 years, at least for a few cancers. With regard to human gene therapy, I think it will take a while. So again, there are some clinical trials that have started, but before to see really the technology being used, I think it will take maybe 5 years, 10 years more. And then only for certain diseases. What is important is to first show some proofs of concepts and then develop further the technology for other diseases and those that are currently targeted, because they are simpler to target.
There are quite a lot of candidate diseases, aren't there? I mean, things like cystic fibrosis, but also some of the very severe immunodeficiencies that are monogenic. You know, there's just the one gene causing them. Those are the kind of areas, I guess, that we might see the first applications. But there's huge regulatory hurdles to overcome.
Exactly, but actually probably, and then the most promising thing what we'll see development of CRISPR will be using this ex vivo therapy, when you can take out cells from the body, do some genome editing in the cells and put these cells back, like stem cells that form blood or something like that, yeah.
Because as we alluded to before, you know, the difficulties remain about how to introduce the altered cells into the body. I mean, that's still a major problem. Let's take another question here.
So I wanted to, I'm Mary Sue Coleman, president of the Association of American Universities. What I wanted to ask you about, though, was just the announcement this week about the Duchenne muscular dystrophy correction in dogs, which was very interesting because they had a model of the disease in dogs that was quite unusual. But it's very much like human, and the idea that the efficiency of CRISPR to get enough cells to be modified so that they could then clear the abnormal protein is quite amazing. And so I just wanted to ask you about that development just this week and what that might mean for a subsequent human, though I know the transition from even dogs to humans may be shorter than from mice to humans, but it's still pretty daunting. What do you think that means then for clinical application?
So the question was specifically about recent reports of using CRISPR technology for effectively as gene therapy for muscular dystrophy models in dogs. Now, I believe, my understanding is that clinical trials using CRISPR for humans with muscular dystrophy have begun in Europe. But have you got any views on that specific example, which is right at the forefront of the potential clinical application?
Yeah, so I think that it shows really huge potential of CRISPR, and this paper just demonstrates that actually you can relieve symptoms in dogs, and that not correcting DNA in all cells, but let's say just in a certain number of cells, and that gives kind of an improvement. So that's really amazing.
Okay. Now, I think there was a question on that aisle up there near Marina.
I'm Silvia Torres. I'm an astronomer from the International Astronomical Union, but I am speaking here as a Mexican citizen. And in Mexico, there's been a big debate now about the genetically modified, for example, corn, that there's a big debate that on two issues. One, that it'll hurt our health to eat that kind of food. And the second one is that it will extinguish the previous crops or the previous strands of variety of corn. So do you have any thoughts on this very complex subject in a sense?
So this is a question about a new Mexican GM modified corn, was it?
Well, the use of genetically modified corn and how it would affect first the health of humans, and secondly the previous strands of corns, that the variety of corns that it would extinguish them.
So this is, I guess, a general question and worry that by using these sort of technologies, we're going to create a monoculture in which we lose all previous varieties of crops and become dependent on one crop because it's got particular qualities, and whether it has health concerns attached to it. I mean, that seems to me to be a perennial problem with any genetically modified food. Those are the things that come up most frequently.
But my point of view is that this has nothing to see with the technology. It's a choice of commercialization of a certain type of crops. Because in principle, CRISPR-Cas9 allows the reverse. It allows to go back to the diversity of crops. So what is discussed here is more with regard to the regulations of what type of crops are allowed to be commercialized. Because one could also think differently. If a local farmer would have the possibility to engineer a crop that would be more adapted to the soil of his or her environment, then there will be a possibility for a local farmer to just create the crop that is more let's say, amenable for the soil, and even increase the local production of a variety of crops that could also go back to the crop that were existing prior maybe to their recent distinction, their recent, how do you say, the fact that they're not produced any longer. So I think it has nothing to see with CRISPR.
And what's for sure is that in many of these big debates about science technologies, it's actually not about the technology in the end at all. It ends up being about emotion and beliefs.
Yeah, and the choice of
the technology. There are all sorts of things which are not about the science at all, which are actually really going to be responsible for whether something is introduced or not. One more question up there.
My name is Stan Browns. I have a philosophical, more philosophical question. I was wondering, so in prokaryotes, this CRISPR system is so abundant, and I was wondering why we don't see any CRISPR systems in eukaryotes, even simple eukaryotes. Can you think of some reasons why these systems wouldn't or don't appear to work in eukaryotes, even simple or more complex eukaryotes?
Well, that's a terrifically science question.
Yes.
As a change from discussions about ethics and politics. So this is to either of you, but I'm going to point it to you, Virginijus, first of all. The question was, if bacteria and prokaryotes have this system, why do we not see similar systems in complex life, the eukaryotes?
Yeah, so it's a question that you expect to get at scientific meeting rather than general discussion. But of course, there are no CRISPR in eukaryotic cells, but actually there are other antiviral defense systems like RNA silencing systems in eukaryotes that are actually similar or analogous to CRISPR, but they work by different mechanisms, use different enzymes, but still,
So they've been superseded by better systems.
Or invented it once more.
But we also know that
independently,
something like 8% of the human genome is derived from viruses anyway, so maybe those systems haven't, in evolutionary timescales, haven't worked that brilliantly.
Yeah, you suggest to look more at the junk, junk DNA.
We're more virus than we are male.
But
Just don't go there. Don't go there, Adam. Don't go there. There was one more question which I saw up there.
Thank you. I'm Brooke Smith from the Kavli Foundation. First, congratulations on not just the award, but your great work. I have a question about collaboration. I think it's fascinating that this goes to 3 scientists from 3 labs in 3 countries. And I'm curious if you could give us a little insight into your own journey to connect with one another, and perhaps some insights into how that kind of collaboration and what nurtures it helped this scientific enterprise, and advice to the scientific community about how we could have more collaborations like that.
Because I think you and Jennifer had a long talk, the long walk, I think she said, when you met at a meeting. Was it in San Jose? And that was, you got completely lost in your conversation.
Yes, so from my side, I started to work on CRISPR actually in 2008, but based on the identification of CRISPR RNAs in this bacterium Streptococcus pyogenes that is actually the source of the mechanism that is used now as a technology. Just because I was interested in understanding better about RNA regulatory mechanisms in this bacterium, so we identified RNAs. Among those RNAs, CRISPR, and another RNA, tracer RNA. We worked for a while on this tracer RNA, where we thought that this tracer RNA had another target than CRISPR RNA. Ultimately, we found out that actually tracer RNA was a second RNA of the CRISPR-Cas9 system. And so we published a first article in Nature. And then following this publication, I met Jennifer Doudna, indeed. And so I was interested at the time, we were following up in my lab to try to show that this Cas9 is guided by the two RNAs to cleave DNA, but I was interested in the structural biology aspect of the system. And Jennifer Doudna is a structural biologist, and she was working already on other types of CRISPR systems. And so she listened to my talk, and so I said that I believe that this specifically simple mechanism had a lot of potential, and that we were continuing to show that indeed it cleaves DNA with these two, that Cas9 could cleave DNA, and so we decided to team up, because there was a part of biochemistry and then the part of the structural biology which I wanted to have, let's say, in the hands, because we were already in the lab understanding that there was a diversity of CRISPR-Cas9 systems. This was already shown by colleagues of ours, but that there were different CRISPR-Cas9 systems that were shorter, longer, and with different, it's a bit complicated, but a different also types of recognition of the DNA. I don't enter into the details, but we had already started bioinformatics on the subject, and so it was important for me also to follow on the diversity. So studying the mechanisms and the diversity, so this was a collaboration that lasted for 1 year, and the chance that really we had is that my student and her postdoc got along very, very well. And this is important because it's not always the case. We often want to collaborate as scientists, but really the people doing the job at the end of the day in our labs are young scientists, and it's very important that they can connect and work well together. And this has been the case for this collaboration. So really extremely talented young scientists who have connected very well.
But it's interesting, isn't it, that even in our very connected world with the internet, actually those key collaborations take place in the coffee room on the edges of conferences.
Yes, I mean, there are different ways. I mean, I have had collaborations where I really contacted the person by phone, contacted the person by mail, took the chance that there was a meeting and ask whether there would be an interest. So there are multiple ways, but this is where conferences are nevertheless important, because it's about also having the chance to figure out whether there will be a chemistry and the same way of handling the collaboration. And at least for me, what was important is to go fast, because the competition was tough.
And Virginijus, when you saw that paper in 2007 and you decided that you wanted to be involved in it, you just emailed the person, didn't you?
Just emailed asking for a strain, and actually I was working on CRISPR, starting to work on CRISPR that was identified in a different bacteria. Bacteria is called Streptococcus thermophilus, and it's used for making cheese and yogurt. So I asked for this strain that contained 4 different CRISPR systems from Danisco company at that time. And after some time, they sent me a strain, and then I thought, okay, so we have another problem, because we soon will be deep in yogurt, and what to do? What to do with that? So therefore, we decided, okay, we'll switch to E. coli, and that's how we started our exploration of CRISPR systems. But I fully agree that actually all my most important collaborations actually were started in a conference bar. So when you talk with people over
where it
the
happens.
beer,
So, ladies and gentlemen, we now understand that actually conference bars are absolutely
where all the action happens.
Essential to collaboration.
I'm sure that you have more questions, and our laureates will be sticking around for the rest of the celebrations for the week, and Jennifer Doudna will be joining us later today. But we've run out of time for now, so please could you put your hands together and say congratulations. Emmanuelle Charpentier and Virginijus.